c'est all.
Un laser ici.
Okay, so I'm going to talk in English because I've been asked to talk in English, but I'm very happy if you want to ask questions in French. I can reply in French too. So, just rapidly, I thought I'd give you a bit of my personal history so you know where I come from. I studied psychology and physiology when I was a student in Oxford, and I did my doctorate with Edmund Rolls. This was recording single neurons in awake monkeys, and we recorded, when I was doing my thesis, a whole load of areas, including particularly the orbitofrontal cortex, but also striatum, parietal cortex, lateral hypothalamus, amygdala, hippocampus, supplementary motor area. Not bad for 3 years. It was quite an interesting experience to just sort of record from a wide range of areas. After that, I spent a year as a postdoc in Canada, and I was trying to test Hebb's hypothesis. Donald Hebb, who was actually still alive and was he was very old. He was in Halifax at the time, and so I wanted to test his hypothesis. Which is that you can strengthen synapses by co-activating the inputs and outputs. I spent a whole year getting negative results. I essentially proved that the anaesthetized cortex of a kitten, of a paralyzed and anaesthetized kitten, is not plastic. There we go. So I failed to prove Hebb's hypothesis, unfortunately. Then I went to Paris to Michel Imbert's lab in '83. And there I tested Hebb's hypothesis with Yves Reignac and Elie Bienenstock, and we actually managed to get the neurons to change their properties by actually artificially activating cells while we were presenting stimuli. So that was quite nice. And then I was also working on the dynamics of neural responses in monkey visual area V1 with Simon Celebrini and Yves Cotter. And then in '93 we all moved, well not all of us, but many of us moved to Toulouse to create the Centre de Recherche Cerveau et Cognition, and that's where I've been ever since. And I've been working a lot on what I call ultra-rapid visual categorization and trying to get constraints on how long it takes the brain to compute various things. And in parallel with that experimental work, I've been developing spiking neural models to try and understand how brains can do computation very fast, even with very, very slow hardware, because essentially neurons are really quite slow compared with electronics. And that, so I'll talk about that a bit, so spike-based image processing. And we actually set up a company, SpikeNet Technology, in 1999, which still exists, even after 13 odd years, which develops software which is sort of inspired by how we think that maybe the brain processes images. So, by the way, please do interrupt me. I've got plenty of things to fill up 2 hours, but I'm quite happy to chop bits out if there are things that you want to talk about. That's what we're here for. You've got the overheads, most of them in the book. So just feel free to stop me, and we'll talk about anything you want. So I'll start with a bit about ultra-rapid visual processing. This has been one of the themes in our lab for many years now. I'll talk about spike-based processing, the idea that you can use waves of spikes. Oh, yeah.
I can. Ooh, wow. Is that better? Is that better with the microphone or?
Yes. Yes. Okay. So using a wave of spikes going through the visual system as a way of processing information. So it's an alternative to the more traditional idea that neurons are sending floating point numbers effectively. They actually send spikes, and when you use spikes, there are lots of different ways you can use them in processing. I'd like to get on rapidly, though, to a question that's really been interesting me a lot. How do we learn to recognize stimuli? And I'll be talking a lot about spike time dependent plasticity. This is a learning rule which changes the connection strength. It's actually very similar to Hebb's hypothesis, but it's specifically based on relative spike timing between inputs and outputs of neurons. And I'll argue that this simple rule logically leads to neurons becoming concentrating. That makes the neurons become selective to things that get repeated. And I'll argue that a few tens of presentations of stimuli are enough to get neurons to become tuned. Here's a problem which I'd like to get your opinions on. How do we remember stimuli over very long periods of time? Essentially, I think we probably all had the experience of seeing something or hearing something that we haven't thought about or heard or seen for maybe decades, and yet be able to, oh, that's that colouring book that I used to have at school. Something that you haven't thought about, and yet your visual systems and your auditory systems are capable of telling you, yes, you know what it is. How does the brain do that? You're storing memory traces which can last a lifetime using neurons where we know that in the synapses, the figures are quite amazing. The proteins that make up synapses are being replaced continuously every 12 hours. You're renewing proteins in the synapses. How on earth can you get a system which is having to be rebuilt, continuously to actually store something which is still there, a lifetime later? That's a real challenge, and I'm not sure that many people have got solutions to that. I'm going to propose a fairly outrageous claim, which you are invited to criticise. But the idea would be that if you buy this spike time dependent plasticity idea, and you make the neurons sufficiently selective that they have no spontaneous activity, they will not change their properties over very long periods of time, even if you don't represent the stimuli. Now, that I think would work, but it does involve two extremely controversial hypotheses, which I think many people would just not buy, but I'm going to try them out on you. One is that we have grandmother cells, that's to say cells which only respond to very specific things and are totally unreactive the rest of the time. And the other idea is that we have what I could jokingly call neocortical dark matter. That's to say the idea that maybe a large proportion, maybe 90% of the neurons in cortical areas never fire spikes normally, and you can't record them. It's just, there are reasons to believe this may be true. We may have completely missed one aspect of the brain. We tend to think that we know what happens in the brain when we present visual stimuli and sounds and all the rest of it. But it's not at all inconceivable that right now there are, in your heads, 90% of the neurons in the cortex simply hadn't fired a spike at all in years, maybe. Okay. Very hypothetical, but I hope to convince you that this is actually not maybe so wacky as it sounds.
But then, what is the conceptual difference between the two? Between the grandmother saying and this dark matter?
Well, you could. I mean, the idea that you can have grandmother cells, but you don't have to buy the idea that there are large amounts of cortical machinery that never fire spikes. I mean, you could have one grandmother cell out of every 100. What I'm saying is that actually, potentially, 90% of the cells are actually grandmother cells. Right? Okay, so they're two separate ideas, but we'll see about that. Anyway, so. Hopefully that's got you. Jan's already reacting. Good. Off you go. What's wrong with this?
Well, so it's the ultimate sparse coding, right? That's
super sparse coding. Exactly.
Right. Or k-means coding, right? You have basically one prototype for everything you need to remember. The problem with this, of course, is that it's exponentially inefficient in terms of hardware. When you have only one neuron active out of a very large population.
No, I wouldn't say. No, I mean, you've got the other 10% of the cells which are doing. Not every cell is like this. It could be a large percentage, but you can have your cake and eat it. You can have a nice distributed code plus lots of interspace with lots of other cells which are actually specific. So no, I'm not saying one size fits all for coding. No, you don't have only grandmother cell coding.
You can have everything.
Sure, but for the population that will, the subpopulation that use grandmother cell coding, then if you want to encode n bits, you need 2 to the n units. You need 2 to the n neurons, essentially.
If I want to recognise 100,000 objects using grandmother cell coding, I'll need 100,000.
That's right. And if you allow half of your neurons to be on, then you need log base 2 of 100,000 neurons.
You can code, but you
right?
ASCII is
then
very
you have
good.
everything in between. So
Yeah.
if you allow 2 units to be on, then you need square root of 100,000 neurons to represent it. Because
I'm not going to argue that ASCII coding wasn't a good idea. I mean, you could have a code for the alphabet where you have 127 different neurons, one of which is on,
Right.
And that's not how we store stuff in computers. But
right. So you have a spectrum between sort of a 1 of n code, which is exponentially inefficient, and then the completely dense code, which is as efficient as you can.
Yeah.
But
And
then not
maybe
very
the brain
good-
has all of those at the same time.
Yeah, but then it's not very good for computation because every function is going to be nonlinear in that space, whereas in this other space, every function is going to be linear.
So
But you have everything in between, and that's kind of where interesting things happen.
I agree. But let's come back to the question of, can you store things in a Hopfield net or an ASCII code and hope that you can write new stuff into it and not overwrite the old stuff? That's my problem. I would claim that since I hopefully will convince you, I can make you a grandmother solve for any arbitrary stimulus, and it will never fire to anything else if I want to. Is it useful to do that? Yes, I would claim, because it gives you a way to store the memory for that stimulus in a totally robust, and you can't interfere with it. It will never get wiped out. The cell will just be sitting around waiting for that particular stimulus to come back on again.
And the only proviso is that it's going to cost you an exponential.
It's going to cost you neurons, but we do actually have quite a few of them. So the question is, do we have enough to keep remembering new things every minute for the rest of our lives? Probably.
So the robustness of that stimulus or grandmother cell also depends on the input, right? The input should also remain stable in order for the memory to remain stable. So
yeah.
would that be a
Well, actually no, because okay, we could get onto that in a moment. But I'll show you a way of making a grandmother cell, which, if you show exactly the same stimulus as you did originally, would give the strongest possible response. But if you show something that's only 50% match, it will still respond. So you can actually deform your stimuli quite a lot and still manage to get things to happen. Okay, this is looking promising. Good. This is the idea. Right, fast visual processing. This is something Molly Potter back in the 1970s was famously developed rapid spectral visual presentation. She was having to use, in those days, you had to actually hand stick together frames in a film and run it through a projector. But these days it's trivial, but it was actually quite something to show that at 10 frames a second, you actually can spot targets and things like this really quite well. At 10 frames a second, I'm just going to show you some pictures of animals actually at 10 frames a second, and you can get the idea of what it feels like. Contrast isn't very good here, but it doesn't really matter. You get the impression, you saw them, right? I mean, you maybe, if I was to say, tell me what you saw, you'd probably forgotten them already, apart from maybe one or two. But your visual system was doing the job, right? And it got the answer out, and you forgot about it because it wasn't interesting. If you want to actually measure... I missed out a slide. Oh. Sorry. Okay, so what I should be showing you is, I can explain this one. We did experiments on go-no-go categorization where we flash up one picture, not an RSVP stream, and you just simply ask people to say, is there an animal present? And what we found was that humans can do this pretty well. It's a reaction time distribution for letting go of a button if you see an animal in a picture. The red bits down at the bottom here are the errors, so you're getting 94% correct. Monkeys can do this a lot faster than we can. So this is the human stuff, and this is the monkeys. So we can't get our hand to move in less than around 250, 270 milliseconds. Monkeys can do it in 180, and the mean is a lot shorter. And the performance is actually very similar. I mean, monkeys can do animal detection pretty much as well as we can.
They need a little bit of encouragement too, when you show them pictures of animals like giraffes that they've never seen before. But they actually cotton on pretty quickly, and can add new exemplars into the big category of animals very very efficiently. Why are monkeys faster than we are? Well, it almost doesn't matter what task you give a monkey. You will be 50% slower than him, almost for anything. And I think the rule could be simply related to the fact that monkeys have got small heads. The fact is that conduction delays within the brain are extremely important component of any reaction time. The axons going from V1 to V4 and to IT and all the other things only conduct at about 1 to 2 meters per second. It's very rare to find cell axons within the brain which are faster. So going from here to here, let's say 15 centimeters, that's 150 milliseconds of conduction delay. That's terrible. And so actually Nancy Kanwisher actually never got published, but did a sort of plotted N170 latency as a phase selective potential latency as a function of head size, and you ought to be able to back project to say, well, if you've got different people at different sizes and the smaller heads are slightly faster, you should be able to predict how much computation time it would take if your head had, was a millimetre across. And the answer is it's so close to zero, it's possibly negative, right? In other words, conduction delays could well be nearly all of the computation time. This is encouraging for people like Jan and anybody else who's trying to do this on a computer, because computers don't have a conduction delay problem. I mean, the signals are going around chips at close to the speed of light, if you like. So you could, in principle, if you're doing the same algorithms but in electronics, do this same task in an arbitrarily short period of time. The biology gets away with really crummy hardware. Yeah.
So do you observe the same differences in people with different head sizes?
This is something which has not been properly done. I mean, the fact is that kids on video games, you can't beat them, and they obviously do a lot of practicing. But no, it hasn't been proved, but I would bet a few beers that if somebody actually got around to doing that, they would find a correlation that people with small heads are faster.
So if you look at monkeys versus humans, do you also see other properties which are different, like axonal diameter instead of axonal length, or myelin properties? Or
I think things like the percentage of myelination and the amount of connectivity between V1 and V2, even the numbers of cells are actually quite similar. I mean, the brain, the visual system is surprisingly well conserved between humans and monkeys. They're not fast because we've got a very inefficient system or something like that. No, I think it is probably that. Right. So if we take monkeys, for instance, we're flashing up a picture here, and they're releasing the button to say there's an animal there. I would argue that since the retina isn't connected to the hand, you have to go through a lot of stuff on the way. Almost certainly you have to go through all of this visual processing sequence here, anterior infratemporal cortex, where you find neurons which are selected to things like faces and so on. In fact, if you train monkeys on almost any task, whether it's judging paper clips or whatever, if you train the monkey long enough, you find neurons up here which will be selected for whatever it is that they've been trained to work on. The latencies here, each stage, we're talking about so this is the fastest latency. It's about the fastest a neuron in IT would respond. This is a typical latency. So at each stage, we're adding roughly 10 milliseconds per layer. When you get here, you can't actually, there aren't any connections either from the top end of the visual system to the hand, so you have to go through maybe prefrontal cortex, premotor cortex, motor cortex, down to the spinal cord now, but you have to do the whole lot in 180 milliseconds, and that's not going to leave a lot of time for doing anything fancy like, 10 iterations of a V4, V2 loop and things like this. Every loop is going to take essentially 10 milliseconds at least. So, I mean, you just will not use loops unless you have to. It's just not the way to get the system to respond fast. So, now, much of our work was done with
button releasing. So we have the subject actually or the monkeys with their hand on a button, put up the image on the screen. If it's a target, then you press the screen or you let go of the button, and we just measure how long does it take to move the hand. But it turns out that with eye movements we can do even better. So here, this is a task that we developed a few years ago now. Fixation point which goes off, flash up 2 images, and you just saccade to the side where the target is, and you can define whatever target you like. It turns out that animals are very good targets. We can do this task very well. Relative to the standard go-no-go task, where manual responses are around 400 milliseconds, with coming in around 300 in this case. If you do the saccade task, we actually sort of knock off half of the reaction time. Basically, we're now down to a sort of mean around 200 odd, and the first reliable responses, let's say a bias in the right direction going towards the animal, starts at around 120 to 130 milliseconds. In the original experiment, we just flashed the images onto 20 milliseconds. It turns out you don't need to do that. It's more sensible to do the experiment with leaving the images on for 400 milliseconds. Otherwise, you just remove your stimulus. And under those conditions, and also you'll notice we're not using black background anymore, we use grey ones. That actually works better if you want to get the shortest reaction times. You get, maybe because you're getting a sort of push-pull effect, the black bits and the white bits are both drive in the visual system. And when you do that, we get an even sharper reaction time distribution, mean 180, and again around 120 to 130 milliseconds the first responses coming through. Remember, this is a task where you've never seen the picture before, and you have no advanced information about what sort of animal to look for, how many there are going to be, what size they are, what lighting conditions, and all the rest. The visual system is doing the whole works, and it's getting a behavioural response out in as little as 120 milliseconds. But if you thought that was fast, try human faces. Human faces produce even faster behavioural responses. So let's just try it. This is supposed to go left there and left again, right there. Just imagine I was recording your eye movements, and we were trying to get a measure of how long it takes you to do this. These responses are really very reliable, and the reaction time distribution, this is the 16 subjects. Virtually all the saccades started between 100 and 200 milliseconds. Mean accuracy 89%. These are the errors. You get a few errors here and there. Mean reaction time 140 milliseconds, and the minimum reaction time here. That's to say the bins where you're getting more good, correct responses than errors, statistically, it's 100 to 110 milliseconds. Now, this is the eye moving, right? Okay, so we have to subtract from that the time taken to actually get the muscles to work and the oculomotor plant and all the rest of it. I think we have to move back at least 20 milliseconds to, say, have a number for how long did it actually take the brain to decide that you want to go left or right. We need to find something happening in the brain as early as 80 milliseconds after stimulus onset. And well, we don't have the really tight data on latencies. These are guesses really of what the latencies would be in humans. Adding 50% roughly to the monkey values. But if we're trying to get responses being driven from around here, we're talking about V2, V4, something like this as being. We don't get to IT. It's not possible. IT, where face selective cells are, hasn't even had a chance to say anything yet, and yet your eyes are moving. More recently, this is it's only been presented at a conference so far. We've been doing an experiment where what we actually do is take a big scene with a human in it somewhere, and then we crop the image so that the face of the person can be in any one of, in fact, 16 different positions, two eccentricities, 8 different directions. The subjects don't know that, and their task on every trial is just simply to saccade to the face. this is what it feels like. So again, imagine that I was recording your eye movements, and imagine how accurate you are on that first saccade. Are you going in the right direction? Do you hit it on the first shot? What we find is when this is an unconstrained position. I mean, before what I showed you, it was left-right, so it's just a binary decision. Here, well, there are at least 16 positions. Actually, as far as the subject's concerned, there are an infinite number of positions, because he didn't know that we've only got 16 places. And that adds about 20 milliseconds to the processing time for generating the saccades. We're still getting 92% correct. That's to say that the saccades are going in the right direction. And if we look at the scatter plots, these are for the 8 stimuli at the larger eccentricity. You can see that these saccades are really well grouped. They're not, maybe a bit of undershoot and so on, but really, really surprisingly well constrained. And here's, for me, this is sort of like a radial histogram of the directions. They're really very good. And for me, the killer, and I was talking with Jan last night about this, I would have thought that if you were to say, this should be getting better as time goes on. In other words, if we were to look at the accuracy of the later saccades, I would expect them to be better, because you've had more time to, average together your activity and do all your voting and all the rest of it. But the fact is, if we pick off just the very fastest saccades, the ones under 150 milliseconds, the distribution is essentially identical to the ones which are produced later on. In other words, the visual system not only knows that there's a phase, it can tell you precisely which direction you're going and how far it is away. And that's at 100 milliseconds. Even the very first saccades know where the phase is. So this is phenomenally fast computing, especially, as I say, we're talking about hardware which is rubbish, basically, compared with electronics. Okay, so now everybody knows that, you can buy any camera you get these days, we've got a face detection algorithm in it, there are little green squares that come up. So it's not as if it's not something that we don't know how to do. There are algorithms that will find faces in natural scenes. They're not that good. I mean, they often may get it wrong, but it's not as good as what we're doing here, I don't think. But it's nevertheless not inconceivable to imagine that we do have, some really, really fast stuff for finding faces. Now, so summary of all that. In go-no-go categorization, we can get responses for animal, non-animal from about 270. Oops, sorry. Woo! No display. I've got the same thing. We had to get rid of that. Display. No, not that. Okay. Oh, okay. We'll have to wait. 270 milliseconds for a manual response. 180 for manual response in monkeys. In the saccade task, we're down to 120, 130 for generic animal detection, and down to 100 to 110 for human face detection. If you try other categories of objects like cars and trees and houses and the rest of it, you can't get down below about 140, 150 for this sort of task. So there seems to be a bit of a hierarchy with some really biologically important stuff getting through really fast, and that's an interesting question of why. Or maybe we can come on to it a bit later. So how is it possible? How can you do this so fast? Well, I won't bore you with a thing on all the anatomy of the visual system, but we know we've got lots of cells. We've got 130 million photoreceptors, 1 million optic nerve fibers, 1 million relay cells, about 500 million V1 cells, 250 in V2, and so on and so forth. All these neurons are firing away in response to the stimulus, potentially. I think, given the speed we're talking about, it looks like it's going to have to be something pretty much like a sort of feed-forward pass through a bunch of filters and so on, which will come as no surprise to people who do the state-of-the-art, like Jan and Shimon Ullman and all these other people who do feed-forward convolutional net type models, which essentially are doing a series of filter functions and then doing potentially more or less sophisticated things at each stage and so on. But no feedback loops involved.
as far as I'm aware, for this sort of categorization problem, there aren't any computer vision algorithms which need feedback. Looking at,
Well, at the high level for scene parsing and things like this, there's a lot of
scene parsing.
yeah,
right,
things
yes.
like context, you know.
Yeah.
Then
You want to
there's-
know where things are in detail and so
Right.
on.
Then there is relaxation,
Yeah.
but for everything up to kind of object categories, no, it's all feedforward.
So I think this is, I'm asking, because we were supposed to answer this question, what does the feedback do?
Right.
Yeah, segmenting scenes could well be a feedback-based thing. If you, once you've found the animal, you want to draw a line around it, and that might be something that it requires.
Well, I'll talk about a model tomorrow that does this without feedback, but the dominant models in computer vision do need feedback.
Okay. More people. So, is feed-forward processing enough? Well, I mean, there's this pretty amazing paper by Thomas Seld, and Tommy Poggio, where they actually used, a pretty standard feed-forward model based on the visual system, and they actually compared the performance of their model with on precisely the task that we've been using, which is animal detection. And they used natural distractors and artificial distractors that would roughly match the scale. They masked the stimuli, and here's the performance as D prime for these different size stimuli. And basically, the model is doing as well as humans. So, in a masked situation where you can't sort of think about it particularly, the feed-forward models can get pretty damn close to what humans can do. So, they're getting about 80.
Yeah, but they degrade the performance of humans by showing.
By masking it.
Yeah, but masking it for a very short time. So that's how they.
It wasn't that short, actually. The ISI is, there's a 50 millisecond delay between the onset and the mask. So.
Sure, but if you allow the human subjects to look at the images as long as they want, then the performance is.
Yeah, absolutely. I mean, human. If they're allowed to look around the thing and find it, they'll get 99.9%. Yeah, no, but if we try and just pick off the feed-forward bit, the models are doing pretty much what you can get out of a human, in fact. So that's fairly impressive. So you could say, is that the end of the story? Well, I would say not, because it's missing. These sort of models have one feature that I think is really quite important about how nervous systems transmit information. They have spikes, and I would argue that although you can get the thing to work perfectly well, I don't know how long Tommy Poggio and Thomas Serres thing actually takes it to run on a computer to get 86% correct, whatever it does. I'm sure if you throw enough computers at it, you could get it to do it in half a second or less. But it may be using far more computational power than we are allowed to allow ourselves in the case of a human processing. And I think one of the things which is missing from most computer vision approaches is the idea of spiking. Because real neurons are not sending floating point numbers, they're sending spikes. And that opens up a whole range of options which I think are worth exploring. So for instance, here, this is an idea that we've been working on for 20 years basically, that if you've got a bunch of receptors, these could be in the retina or in any sensory system, if you put in if you flash on an image or whatever, those cells will maybe all fire one spike, so they're all firing at the same rate, one spike per image. And yet, because the ordering, the temporal structure of this wavefront is extremely interesting, it's very rich, and you can learn an awful lot simply by looking at which of these neurons fires first and in what order, without having to count spikes or measure firing rates or whatever. And hopefully convince you that there's other spin-off things which are good for this, because it makes learning easy, and it makes hardware implementation potentially very easy. There are sort of moves afoot to design hardware which will actually do this, which will actually have pulse-based processing and all the rest of it, pretty much like neuron-inspired spikes. The other idea that I'll be talking about is spike-time dependent plasticity. This is the idea that you strengthen connections or you depress the strength of a connection depending on whether that connection was active just before or just after a postsynaptic spike.
And we'll get to that in a bit. In the meantime, let's just sort of say the general principle is that any neuron, if you think of it just like a capacitor which is being charged up, if you put in a stimulus with a relatively weak stimulus, it'll take longer to reach the threshold, and if you increase the intensity, it gets to the threshold faster. So, in a sense, every single neuron, spiking neuron, is a analog to temporal converter, because it doesn't matter what you put in, it doesn't fire spikes at a fixed delay, it fires spikes at times which depend on how good the stimulus is driving the cell. And that means that you can actually convert an intensity profile into a wave of spikes. Here we've got a bunch of spikes, you can't really see the stimulus here. And you could use the precise temporal delays here, but actually there are things you can do even cheaper, like looking at the ordering. So here for this stimulus it's B, A, C, E, D, and for another stimulus it might be E, D, C, A, B. And just looking at orders is already incredibly rich. That's just with 5 neurons you've got a factorial 5 orderings. With 16 you've got 10 to the 13. With 100 you've got it's just incredibly rich. Even under conditions where every neuron fires one spike and therefore the firing rates are unusable. But you can also you can potentially use the ordering information in a very intelligent way, because this is a study. This is getting old now. This is with Reuven van Rooyen. We just took a toy retina, which was just doing little on and off center convolutions on the image at various scales, and we said, let's suppose that each of those little cells is like a little capacitor charging up to the threshold, and they fire a spike after a delay which depends on how well the image is matched to its receptive field at that location. And then we have all those cells sending spikes along the optic nerve to the brain. And we're saying, imagine you're in the brain and you're listening to these spikes coming up. What can you tell? Can you reconstruct the image without using firing rates, just by looking at the ordering? And this is what we did here. So this is reconstructing images on the basis of just the ordering, just who fires first, essentially. Now, down here we've got the percentage of cells which have fired so far, and it's sort of what we can. I mean, if I stop here, for instance, let's say I've stopped it quite early. Can anybody see what that is? A cat.
Lion.
It's not actually a cat. It's a fox. So between 0.2, where people were saying it was a cat, and 0.3% of the retina having fired a spike, we get from animal, mammal, cat maybe, to fox, because you didn't quite have the little points on the ears and so on. And what we find is that this way of reconstructing images based uniquely on the ordering is actually very, very rapid, because essentially any image done like this, by the time 1% of the cells are fired, you can tell me almost certainly what the image is of, and recognise several objects in it and so on. So there's actually a trick which I haven't told you, is that when the spike comes up from this retina, and we're actually, what we're doing is we start off with a grey image, right? Start off with a grey image because we don't know anything, and as spikes come in, who had just a few on-center things and maybe an off-center one on the left, with a low spatial frequency. As spikes come in, we're giving them a weighting that depends on their order. So the first spikes that come in have a very high weighting, because we know that statistically they're the ones that are being most strongly driven. And in fact, the actual lookup table, if you like, we just determined it by taking lots of images, measuring, saying that the first cell that fires will give it a weighting of 100%. By the time, this is rank, log of the rank, so by the time 0.01% of the neurons are fired, the average energy is down to 50%. By the time 0.1, 1 in 1,000, by the time 1 in 1,000 cells are fired, we're down to 15%. By the time 1 in 100 cells are fired, we're down to 3%, which basically means that because statistically it's only the first 1% which is carrying the key information, you can almost forget about the other 99%. If you wanted to build a good, a high efficient thing which is going to use the minimum number of spikes, you could actually say, oh, give me 1%, forget about the rest. Most of the time I don't need it. So you make very sparse codes using this method of ranking things in the time domain, essentially, and then cutting off when you've got enough to recognise. You just let the thing keep going. If you haven't worked out what it is yet, just keep going. When you get there, you say, thank you very much, let's do the next image. And in a sense, it's actually not unlike sort of JPEG and other types of compression things, schemes, where you do lots of convolutions and lots of processing, and you throw away all the low-valued coefficients. You just leave the ones that have got the most weight. Except that here we're sending the information in the order of the weights. So it's like you do your JPEG and you send first the highest value, then the second value, and then the third value, and so on. And that sort of guarantees that the system's going to be as efficient as you can make it. Right. Now, in this sort of thing, if you want to make neurons selective, here we've got a cell which has got a weight set here. These are the synaptic weights, and here are a bunch of cells in input A, B, C, D, E, and they're firing the spikes in this order, which corresponds to the intensity profile here. Now, if this cell was on its own, it wouldn't actually matter what order these cells are firing. You'd get the same sum at the end. It would be this big one plus the next one plus... it doesn't matter what the ordering is. But if you add a little feed-forward shunting inhibition circuit, which exists in all sensory systems, this little inhibition circuit is going to turn down the gain, the sensitivity of the cell. So before any cell has come in, the inhibition is 0, and the cell will respond maximally. But as spikes come in, it's getting less and less sensitive because the inhibition is shunting the effect. So for this cell, if you want to make it fire best, or most strongly actually excited, you want to fire B, A, C, D, E, because that's the weight sets. And if you've got lots of cells, they can all have different weight sets. So this one here would fire, would get the strongest activation if you hit E, D, C, B, and then A in that order. Okay? So you can make this, with a pretty cheap hardware, you can make the neurons selective to the ordering, and let's throw in one other feature, which is we can have feedback inhibition. These are, we've got these 5 cells here. If we wanted to pick off the most, as soon as one cell is fired, we can use the fact that it's fired to inhibit all the others. So it's going to do a winner-take-all, if you like, in the temporal domain on the input. So this guy here will fire, F, and it can stop anybody else firing. Or it can maybe stop some fixed percentage of them firing. All these things are very easy to do in the temporal domain, much harder to do in conventional computing. So, I mean, if you want to, to put it in the extreme way, if you had 1 million inputs, and then you corresponding to the retina, and you flashed up a picture of the night sky, and you said, I want to know where the brightest star in the sky is. Well, you could measure the grayscale values, and then you could sort them, and do some sequential search on it. Or you just say the first guy to fire inhibits all the other ones, and then you know where the brightest star in the sky is. So this is, I think, what nature does with spikes and the temporal domain. You wouldn't do, or you wouldn't do it in such an efficient way if you just converted everything into floating point numbers and then sorted them and do all the rest of it. So going back a lot, this is embarrassingly old now, but I mean we showed back in 1998 that you could take a really simple architecture. We just have on and off center receptive fields in the retina, a set of oriented filter banks, and then feed these into 3 maps of neurons that we train to respond to eyes and mouths and things like this. And you know, we set up the weights between the orientation maps and the mouth and the right eye and the left eye and so on. And yeah, actually it worked. I mean, it was getting, it wasn't quite state of the art, but I mean, in 1998, but it wasn't that far off. And given that there isn't very much in it, it wasn't too bad. And all done with, you know, no more than one spike per neuron, etc. A bit later, Arnaud Delon, we took an even more stupid architecture where we didn't even bother having face intermediate features. We just went directly from oriented filter maps into face identification maps, if you like. This was with the Olivetti face database, so there are 40 people, 10 different views. We learn one face for each person, and that gives us a sort of set of weights, which produces like a sort of a receptive field for each one of them. And here you can see this face is being shown, and you've got these are the spikes in the retina and in the various oriented filter banks. And this little spike here is saying that the first neuron to fire in the system in this recognition layer was for person F2, which is the right one, even though it wasn't actually the same face as was used for training. And this actually worked. You could throw the entire Olivetti face database in a reconstructed image, and I think there are just a few cases where it didn't tell you which face was which. It was 1, 2, 3, maybe 4, I think, where it said it was a face, it just didn't get the right one. Another couple of points about this is you can lower the contrast a lot and it still works, simply because the ordering doesn't change when you lower the contrast. You can get down to 3% contrast and it's still working. And you can randomize pixels. 50% of the pixels here have been given random values and it still works. So pretty robust system. And it's just based on the first spikes and the ordering of the first spikes. So that's what we, trade secrets a bit, but this is what we put in SpikeNet originally when we set up the company in 1999. The training algorithm puts high weights on the early firing inputs and zero weights or zero low weights on the later firing inputs. And we have the tricky thing is setting the threshold in the recognition layer so they're all equally balanced. That's a little bit of a fiddle. So, in its first iteration, we actually stuck the recognition layer immediately after the orientation filter banks. That's not how the brain does it, but it could do. It's just very expensive in neurons. You have to have at every single point in every image, you have to have 40 different neurons to code to 40 different people. And you couldn't do that if you wanted to do this in the brain. You have a V2 that was 40 times larger for doing 40 objects. That won't work. Nevertheless, that thing actually has quite a lot of built-in invariance. This is trained on the center image here. Little red dots are the neurons going over threshold, and you can shift the luminance all over the place and blur it and add noise and all the rest of it. Doesn't really matter. And you can rotate by 10 degrees. You can change the size. You can warp it with the perspective, and you can do 3D rotations. And all of that is without doing anything clever. It's just the so clearly this is not realistic. I mean, at the very least, this isn't a visual system. It's more like this. But you could argue that it's like this simply because if you want to recognise 1 million different objects wherever they are and whatever the size, although you could do it by having zillions of recognition neurons very early on, it's more efficient to do it by breaking things up and having the neurons at the top of the system not able to tell you where the object is. They're doing the classification thing, right? So the advantage of this, less neurons. Disadvantage of this, you don't know where the stimulus is, so you have to backtrack, and this is what we were talking about earlier on about a system like this. These neurons at the top will be able to tell you that there's a phase present, but they don't know necessarily where it is, what size it is, and you may have to backtrack to be able to work out, draw the line around the edge of the face and so on. Right, so this is all stuff that we've been working on for 15 years or whatever. Much of it was just pure speculation, really, because we didn't know to what extent these temporal differences are important. I'd done enough neurophysiology to want to do all the experiments to prove it. But I was very happy when Tim Gollish and Marcus Meister published this paper in Science. This is actually salamander retina, flashing patterns. And just look at the variations in latency. This is a flash stimulus, right? So there's no temporal structure to the stimulus. Just the stimulus comes on, and look at how much the latency is shifting. There's a 20 millisecond latency shift, depending on whether you're hitting the neuron optimally or not. And in this figure, which I like, they've taken a natural image of a salamander, and sort of imagine you take one cell and you flash this picture in various positions, and it allows you to, for instance, this line here. These are the spikes that are generated by the stimulus. You can see again, the enormous variations in latency and number of spikes. When it's shorter latency, you get more spikes. So you could say, oh, frequency firing. This would count the spikes, so we'll get a spike rate code. But this is what you get if you reconstruct the image on the basis of the relative timing. This guy is firing before this one. Therefore we make it blacker. And this is what you get if you use what's the percentage now? What percentage of neurophysiologists think that the only thing they have to measure is firing rate? It's still quite a lot. And there's an awful lot of modelers who think the only thing that the neurons have to do is to set the floating point number. That's what you'd get if you were just counting spikes and trying to reconstruct on the basis of that. I don't think it makes sense to say all I want to do is count the number of spikes in 100 milliseconds and measure the firing rate. That isn't a good strategy. As a little break there, I'll just stop there maybe and see if there's any questions. I'm talking a lot, and is that fairly clear? So, basically, I've been trying to argue that spikes change the story quite a lot, because you can do things easily in a spike processing mode. And I think the example of a winner-take-all is a particularly good one. Winner-take-all is a function which is not trivial to implement when you've got lots of data to process. But if you switch into the temporal domain and have neurons firing spikes, and you just say the first guy to fire is the one I'm interested in, then your computational load has gone down by potentially several orders of magnitude.
So would you say the exact latencies are important, or is it just the ranks?
I don't think the actual latencies can be used. If you're sitting in the visual cortex and you have a stimulus comes on, you don't know when it came on. At least normally you don't. I mean, if you move your eye onto a target, I suppose you could use a sort of corollary discharge and so on to get an estimate of it. But I mean, it's just not going to be very easy for you to measure the latency. Knowing that the latency relative to the stimulus coming on was 43 milliseconds. In any case, what's that going to tell you? I mean, if you were to do the ultimate experiment and you recorded from a V1 neuron and you measured its response to every possible stimulus and you knew precisely what you would do, and then I show you, I then flash up a stimulus, you don't know what it is, and I tell you that it was firing at 43 spikes per second, can you tell me what the stimulus was? You can't. I mean, there's nothing you can say. It could be the right orientation and low contrast, or a not very good orientation and a very high contrast, or an infinite number of things. You only can read out the information if you know what the other cells are doing, and that's an ordering code. So absolute latency, I think, you can forget it. I mean, it's not measurable, and you wouldn't be able to know what to do with it anyway. Any more thoughts? Yes.
So you have just presented a spike-based mechanism by which you can decode reliably and very fast the image that has been presented to the system. Is it fast enough to account for the fast processing experiments you presented in the first part of the talk?
I think so, yeah. I mean, let's take the, I showed you the network which did 1 out of 40 face identification things, and it was immediately after the oriented filter banks. And we're only using the very first neurons to fire in those oriented filter banks. We don't have to, it's the first spikes at every stage, and we can tell you the neuron that fired to say it's face F2 had incredibly short latency actually, because it was the first neuron to fire in that layer. So yeah, I think this way of doing things can explain why we can get really fast computations done. Okay, right, recognizing stimuli. So we recognise lots of stuff, including, I don't know, how many people know what this is? Quite a lot. It's a famous picture of a Dalmatian. If you haven't seen it before, then you might be wondering what I'm talking about, but it's an interesting fact that you only had to see this picture once in a Psych 101 thing 10 years ago, and you know what it is. You've stored that basically meaningless bunch of blobs, and you know you can call it, oh, it's that Dalmatian picture. And I bet you could do that even if you hadn't actually seen it since, for several years. So let's see. People who recognise that as being a Dalmatian, can you estimate how long it was since you last saw it? Is anybody here going to tell me it was more than 5 years? Maybe?
Less than 5 years.
Less than 5 years? Anybody with more than 5 years? No, okay, well, yeah, you can imagine it wouldn't be a miracle if somebody said, oh yeah, actually I saw that in 1987, Patrick Kavanagh's lecture, or something, and they might be able to recognise it, and that's what I'm talking about. It's this ability to recognise things.
It was in your lecture.
And it was on Monday.
in your master.
Oh, well, yes, let me just remind, we're not just talking about vision. Who can recognise this?
Beatles.
Thank you very much.
Come on.
Yes, your Beatles help, very good, excellent. What about that?
Yeah.
You obviously listen to the same rubbish as me.
Let
It be. It could have been, but no. Actually, you're recognising the key in which it's played with. You may not be musicians, but actually, if you play that chord shifted up a fifth, then you won't say "let it be." You're actually storing the auditory patterns. So, shut up. So how do we do this? How do we store stuff? I don't know when the last time you heard Hotel California was, but it could be potentially, 1974 or something. You never know if you've been out of not listening to the radio for a long time. I reckon that we can potentially explain this sort of thing with spike time dependent plasticity. This is just the rule that says, if you've got a neuron and it fires a spike and you just had an input that had been active, and you reinforce it, and you get these sort of learning curves we saw earlier on, where this is the lead time. If an incoming spike was just before the postsynaptic spike, then you give it a big boost. A bit less if it's, say, 40 milliseconds before. If it fires afterwards, then you depress it. And a lot of this has been shown experimentally in a number of systems since the late 90s, and loads of people have been interested in this from a computational point of view, lots of modelling and so on. One thing that we think is very important, which I'm not sure that everybody has realised, that it's one consequence of this is that whenever you have an input pattern that occurs repeatedly, high synaptic weights will concentrate on the early firing inputs. And to convince you that this is the case, let's take a really simple case. This is a neuron, toy neuron with just 6 synapses. It's got a threshold of 2, and I've initialized the synapses all with a weight of 1/3. Okay. Now we could use something a fancy function like this, but we'll make life easy. We're just going to have if any synapse active before gets pushed up by 1/3. Any synapse that fires after the spike is pushed down by 1/3, and we're going to present this sequence of spikes. We're going to have these wave of spikes coming into the neuron. So the first presentation, here come the spikes, bang, 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 bang. So we've activated all 6 of them.
you get to 2, the neuron fires a spike, and we reinforce all 6 of them. Okay, so they're now all moved up to 2/3. Do it again. Now you only need these 3 to active. So these 3 now get pushed up to 3/3. These get pushed down. Presentation 3, you only need these 2. So these ones have been pushed up to their maximum. This guy is starting to drop down. These are down at 0. Presentation 4, keep going. And finally, presentation 5. 5 presentations, and I've put my high synaptic weights on the first 2 to fire and set everybody else to 0. Now that's a very simple illustration, just so you can understand there's nothing magic here. But if you do the same thing with thousands of inputs, and you have waves of spikes with noisy patterns, and you keep playing the same patterns over and over again, you get exactly the same thing. The neuron will end up with high synaptic weights on the early firing input. So if this pattern, which doesn't look very structured at all, is just repeatedly presented, a few 100 times, the latency has dropped down. This is latency here, and this is a sort of a map of synaptic weights. The white bits are where the high weights are. It's basically stabilized in a 100 or so presentations. It's found the early firing inputs, and it's pretty robust because you can jitter all the spikes by up to 10 milliseconds, and it doesn't make any difference. It will converge in the same time. If you have more jitter, it will still end up in the same place. It just takes a bit longer. And if you add in spontaneous activity, so on every trial you add in extra spikes, which are total noise, again only slows down the convergence. You will always end up with the high weights on the early firing inputs. And with Tim Masquelier, we looked at what this would do. If you imagine we got, in fact, we had 2,000 afferents, 2,000 axons coming in to a system, and they've all got totally random Poisson processes, uncorrelated, going up and down independently. In other words, it's complete garbage. There's absolutely nothing of interest whatsoever, except that there's actually a bunch of, here, these red spikes. You can't see them particularly well here. We've just sort of selected a subset of the afferents, of the 2,000 afferents, and sort of copied a randomly chosen bit of 50 milliseconds and copy-pasted it at random intervals later on. Now, if I was to say, if I removed the red and said, tell me what repeats in that sequence, you haven't got a hope. I mean, and don't forget there are actually 2,000 of these. Okay, I could give you tons, terabytes of this stuff, and you have patterns occurring, regularly. But finding them would be a nightmare because they're very well hidden. They have nothing special about them. And by definition, we just took random bits of the stimulus. Well, you might think this is a the sort of problem that you give to a computer scientist, and they say, oh, well, it's intractable. Maybe it's not intractable, but it's a tough one. Well, you don't need a computer. You just need one neuron with STDP, and it'll find it for you. Why? Because, well, it would just take an integrate and fire cell. Oops. Integrate and fire cell here. Just nothing special there. STDP, which will reinforce every time the pattern occurs. If the cell fires in the pattern, you're going to reinforce selectively synapses which are part of the pattern, which means that they'll be more likely to fire the cell if they reappear again. And you can see this happening here. So the initial state, this is the first 1 second of activation. We've set the synaptic weights on the 2,000 afferents at random low values. So the neuron is actually going over the threshold a lot. It's firing away quite happily, 50 spikes per second. This is where the pattern starts reoccurring, okay? As I say, we're going to reinforce synapses which are part of the pattern above the other ones. And within 5 seconds, this is 5 seconds later, the neuron, these are the last noise spikes. From then on, the cell is firing 1 spike on every pattern. And if we leave If we leave this for a few minutes, this is now 450 seconds later, no, so what's that, 5 or 6 minutes later. It's now not only responding to the stimulus, it's found the very beginning of that repeating pattern. And it's firing a spike within about 5 milliseconds every time the pattern reoccurs. One neuron, completely noise as the data. I haven't made this problem particularly easy. It's about as hard as they come as signal detection, I would have thought. But there's nothing clever. Let me try and convince you. There's nothing magic here. This is the 2,000 afferents, of which 1,000 here are in the pattern. They're firing somewhere in this pattern here. And the little white dots are just showing the strength of the EPSP, the little synaptic potential that's been produced. You can see that all the synapses within that are not in the pattern have basically gone down to 0, and some of the ones that are in the pattern have got big weights. It's the ones that fire right at the beginning of the pattern. Okay, so here's the membrane potential of the cell, and when the pattern starts, it just sort of ramps up and fires a spike. This within 5-10 milliseconds of the start of the stimulus. There's nothing magic. It's an integrate and fire cell with STDP. Nothing else. If you were to turn the pattern back and play it backwards to front, play the same sequence of spikes but the other way around, it would naturally fire at the end of the pattern, because that's when these particular afferents would be firing. But the interesting thing is that as soon as it fired there, it will start backtracking again and find the beginning. So it will always find the beginning of any pattern that repeats. See what I mean? So now that's one neuron. I think we can allow ourselves a few more than one neuron. So let's have 3 neurons. If you've got 3 neurons listening to the same 2,000 afferents with patterns hidden in them, and we've got inhibitory connections between the neurons, which mean that once one neuron is fired, it will inhibit the other guys, so that they can't fire at the same time, right? But they have to wait a bit for the inhibition to relax. What happens then? Well, we get the following situation. So initially, the neurons are all firing, but by the time they've settled down, you've got one neuron which is firing after the first 5-10 milliseconds, then another one after another 10 milliseconds, and another one after another 15 milliseconds. So if you have lots of cells, they will fire at specific points through this pattern. And so it will be very easy for a later stage to pick up that there's a sequence going on every time. Okay? And of course, if there isn't just one pattern, but you've got several patterns, so here we've got A, B, and C patterns, the neurons will also fight to distribute. It's a competitive learning mechanism where different neurons will learn different stimuli spontaneously. No reinforcement, it's completely unsupervised learning. If we take one of our cells and we put it at the end of our toy visual system that the retina that we already seen, and then just keep flashing the same stimulus. So you've got one neuron, the brain has one neuron and it's listening to the retina. What's going to happen? Well, a little bit complicated, but let's see. So let me just stop this and try and explain what we've got. We've got here, this is the stimulus that we're flashing over and over again. Here we've got the weights from these retinal ganglion cells to this cell here, and they've been ordered they've been ordered according to the rank of when they fire for the stimulus in question. This is the membrane potential of the cell. So at the beginning, it takes all the weights are low random weights, and it takes 190 milliseconds to get over the threshold. But if you keep flashing the same thing over and over again, you get this wave of potentiation going through, and it's tracking. All the weights are going to end up concentrated on the very first ones to fire. The cell now has a really short latency, and the other thing that you can note is this is a reconstructed receptive field, which initially is just noise. And during the learning process, it's incomprehensible. It's just, you can't work out what the stimulus is from this. But when it gets to the end, it's got a receptive field which matches whatever it was that was being repeated. And if you do that with a cat
picture. It's exactly the same thing. Everything gets faster, and you end up with the weights that are retained are the ones that fired early, so we picked off the whiskers and the eye. And if you do it with a Mona Lisa, then the same thing. You end up with a receptive field which has, roughly, it's a retina's eye view of the Mona Lisa. It's not a fantastic one, but it actually is very selective. If you don't show something which is very close to the Mona Lisa, the cell will not get over threshold. Now, obviously, we don't have just one neuron in the brain. We have lots of them. So, you have to imagine that this is going on all over the place. And we've done a slightly more sophisticated job here in this other paper, where we took a more sensible thing, where there actually are some neurons in the previous. It's not just a retina connected to one neuron. We've got three maps of cells at the top end of the system. Top end of the system here. We've got three maps of cells with weight sharing, actually, and we're going to show pictures from the Caltech face database. So we've got green cells, red cells, and blue cells, and a couple of rules. Only the first green cell is allowed to fire, and only one green cell, only one red cell, only one blue cell, and they're not allowed to fire in the same place. Now, initially, you can see the thing is going off pretty much at random, but by the time we've presented about 100 stimuli, we're up to 83 now, we're starting to see receptive fields forming in these neurons, which are, starting to get useful. So, for instance, let me stop. Here, for instance, we've got the red cell has got a receptive field which is looking a bit like the top half of the face. The blue cell, the bottom half of the face. The green cell, well, it wasn't very obvious here, but it's actually tending to fire on the left eye and the nose. And we've only shown 100 things, and it's already doing pretty good as a face detection system. It's actually hitting, there are occasionally misses, like that one, for instance, but it's doing pretty well. This is totally unsupervised. Nobody said anything about faces. The only reason why it's learning about faces is because it's all Bayesian, this. What's the most likely stimuli in the environment? Well, in this data set, it's faces. I mean, if every one of these faces had been shown on a scene where there was a clock in the background, then the neuron would have learned about clocks, maybe. But it's just what is the stuff which is being shown, which is the highest frequency in the environment, tells you what you're going to learn about. If you do this, we did this with a Caltech motorbike database, and you get bits of motorbikes. And if you have a database which has 1/3 motorbike, 1/3 faces, and 1/3 other random things, you get some motorbike bits, some faces, and so on. I mean, it just dispatches the selectivity of the units between whatever it is that you're showing. We've been playing with these ideas recently. This is Toby Delbrook, and he's made a chip which is a bit like the retina. It's certainly more like a retina than a normal camera. It has 128 by 128 pixels, and for each pixel, just a little circuit which detects whether the luminance has gone up since the last spike. If the luminance goes up by 10%, it generates an event, which is essentially an on spike, okay, for that pixel. If the luminance drops by 10%, it generates an off spike. And every time you generate a spike, you save the current luminance level. And this produces an asynchronous chip, which produces this sort of stuff on the output. Lots of spikes coming up. And it's just pure luminance. It's not quite as sophisticated as a real retina. But it's very rich. You've got lots of stuff here. And this is actually cars going by on a freeway in Pasadena. So this is very heavily structured spatiotemporal patterns of spikes. Let's see what happens if we feed that into a competitive STDB-based network. And this is what we've done with. So Olivier Bichelet, who was just finishing up his thesis, did this with these other guys are from in Orsay and Saclay in the Syrah, and we're using these sort of things, and so we took
We didn't have, at the time we did this, we didn't actually have one of these chips. We just downloaded the data off the web. They have a sort of a 90-second sequence of cars going by on a 6-lane freeway in Pasadena. So we got the data and then fed that into. Oh, I need to show you the architecture. It's not there. Sorry. Why can't I see it? It's in your figure. It's important that you know what I'm talking about, so let me use. Basically, we just take there's 128 by 128 times 2, because you've got the on and offs, and 32,000 spiking neurons in the input. And here's the architecture. It's just this is an event address event representation. They just send numbers through the spikes, going up to the optic nerve, if you like, and every one of these projects to every one of these 60 neurons here, which all have this lateral inhibition thing, so they're not allowed to fire. They can't learn the same thing, and then those cells project to a second layer of 10 neurons, also with interconnection. And what do we find? Well, after we took the 90 seconds of stimulation and ran it actually, I think, 3 or 4 times. So we're talking about 5 or 6 minutes of experience. And the layer 1 cells have learned to respond to cars going by at particular points in the image. Why? Because those little packets of spikes are highly correlated. You get a bunch, and the neuron will learn to pick up in each case, as cars go by particular places. Those are you won't get a cell responding to 2 cars going by unless they always drove by in parallel. No, they don't. They're uncorrelated on the different lanes, and that's why each neuron has picked up a different lane and a different position. But for me, the really impressive thing is the second layer neurons, which are actually learning sequences in these things. The 10 cells, they each picked a particular lane, and we were effectively counting cars going by on the lane. You can see this here. This is the 6 lanes, and these are reference events, hand-coded cars going by. And for each one of these sequences, we've chosen the best neuron out of the 10. And you can see that I think got 98% correct of it's learned to count cars, and nobody told it anything, right? I mean, the only thing we did was just dump loads of all this data, and the first layer made receptive fields for cars going at different positions on the different lanes, and the second layer ended up counting the cars, and nobody told it anything. It didn't know what the task was, it didn't know what the data was, it had no knowledge whatsoever. So this, for me, is suggesting that the power of this sort of architecture to learn about things which are going on in the environment that repeat is quite remarkable. And I would like to suggest that, if you imagine the same architecture, I mean, there's nothing special about these neurons. We could plug in anything you like. It can be dolphin cries, it can be electromagnetic stuff, electric fish, tactile sensors, visual stimuli, obviously. Every time there are things that repeat, the neurons will become selective to those things. I would claim. There may be a little bit of tweaking to get the time constants of the inhibition and the time constants of leakage to be optimal for a particular problem. If you're doing speech detection, you might not want to do the same time constants as if you're doing tactile recognition or something like this. But for a particular sensory domain, there'll be a few things that you have to set up so that it works, I suspect.
So this system is doing this without it's learning without supervision. It's
Totally
learning
unsupervised.
without feedback,
Without feedback.
without any principle of Bayesian inference or something, reconstruction of.
Well, actually, I'm not sure there's no principle, because it's actually working, it's interesting. What the neurons do depends on the statistics of what comes in. So if, one of the cells actually ended up being looking as if it was interested in lorries, trucks going by. But there weren't enough trucks to make it worth it. So, but if you had 50% trucks and 50% cars, then I would predict you would get truck detectors as easily as car detectors. I mean, it is doing, as far as I can see, it's learning about the statistics of what things are the most likely in the environment, and it will code those events specifically. And if you're in an auditory environment where people speak French, it will learn about French phonemes. If you're in an auditory environment where you're learning Japanese or English, then you learn about those, and then you've got a very biased auditory system within minutes, in a very young kid, because this is all going very fast. Minutes will do it, and you're already.
What I'm surprised at, and at the very least, it is doing so. The learning does not involve explicitly any Bayesian type of inference. In the learning rule, in the STDP learning rule, there's nothing like that. That's just.
I think I should stress. None of the things that we put into this are anything that would shock anybody I know. It's just leaky integrated fire and a simple STDP rule. So no, we haven't put anything clever. We haven't designed into the system anything like Bayesian inference, and yet it seems to be doing something like that. It's not, we haven't done deep belief networks, but the units that get formed up spontaneously, could be thought of as doing something quite profound. It's working out that there are events that correspond to cars going by on autoroutes, with no a priori knowledge. I mean, that looks pretty deep in what the system is generating as representations.
Sorry, my question was, do you think there's a chance that adding feedback connections in this system will mess the STDP learning?
No, actually, STDP learning doesn't care whether the inputs are coming from the retina or from elsewhere. So if you imagine a V1 cell, which is learnt about orientations, an orientation edge detector based on what it's learnt about things that happen in the retina. Okay? And let's suppose that that cell also has connections coming back from MT, where the neurons code motion and so on. Let's suppose that there are MT neurons which are coding a movement like this. Those cells will predict that this cell will fire. So the cell could perfectly well connect up feedback connections from MT because they predict that the cell's going to fire to the bar when it reaches here. So it's not just for feed forward. All neuron, let's push it really far. Every cell in the brain could be thought of as trying to predict its own spikes using whatever's coming in. And that stuff coming in can be directly from the environment, via the retina or the auditory nerve, but it could also be by prediction from frontal cortex or wherever you like. If those predictions are able to say that this cell is likely to fire in the next 20 milliseconds, then it will get connected in two. It's already pretty spectacular what it can do just on the basis of driving it with the sensory input. Imagine what's going to happen if you start getting the system thinking, about what things are likely to happen. Or, the sky's the limit, that seems to me. So no, it's not just a feed forward. So, summarise that. STDP concentrates high synaptic weights on early firing inputs. Inhibitory connections between the neurons make a sort of competitive network where neurons are fighting to find something which hasn't already been coded by the neighbours. If you've got a system where you've already coded all the things that
coming in, the car's going by, and then all of a sudden somebody starts driving in the other direction down the autoroute, then there aren't any neurons that can detect it. Nobody fires, and then it's up to everybody. There's nothing to stop other cells learning this new thing. So the system will not learn as long as there are some cells in the system which are saying, okay, now I know what that is. But as soon as you don't know what it is, the inhibition disappears, and cells are now free to start looking for new patterns which somebody else hasn't picked up. So this will automatically start reacting to a new thing, a new letter shape or a new whatever, which you don't know about. It will start learning about it. Different neurons respond to different bits of the same temporal sequence.
One neuron for each input, for each object?
It can be. It depends on the receptive field size of the things. I mean, in the case of the cars going by on the autoroute, you have one neuron for each location in the image. But it doesn't have to be. I mean, you can also code the car with oriented edges and all the rest of it, and then you have lots of cells. I didn't mention this, but you can actually, this is the sort of circuit we're talking about, could be done in hardware, and that's actually Olivier Bichelet's, the other half of his thesis is on, testing components like NOMPETs and other weird things which you can do STDP-like learning potentially with. You can build a circuit which does this. You've got a paper coming out in neural computation on doing Pavlov's dog. It's essentially a neuron with two inputs, one for the bell and one for the food, but it's actually using a weird semiconductor device called a NOMPET, which I don't know anything about, and it does this, and you can do this with a device which is a physical instantation of something like that. Right. Give me a time scale for finishing off.
Sure. Take your time.
I don't want to overdo this. When did I start? Was it sort of 1:30?
We were half an hour late.
Half an hour late. Okay, so even without exaggerating, I do actually have another 25 minutes or so, right? Is that okay? Is that okay with you lot? Or do you want? I mean, you can have a break if you like. I mean, keep going.
Do you need a break? Do you need a break a little bit?
Do I need a break now?
Yeah, just for water or something.
Water, yeah, that would be good. So I've been telling you about this STDP learning thing and saying, and it looks, I think it looks rather plausible, right? Is it possible that...
Were you the first talking of STDP?
Oh, no. Those of you who talk about STDP, I think what our group has done, which is original, is just to say, well, STDP in the context of temporally structured spiking, the weights focus on the early firing inputs, and that's the new bit. I mean, but there are probably 1,000 papers modelling STDP. But generally people have been started from the notion that neurons are sending things in a firing rate code, and so there's all piles of stuff showing you can get neurons to do this, that, and the other on the firing rate. That's not how real sensory devices send information to the brain. They send it as very richly temporal patterns.
But learning STDP.
Learning. Well, STDP is learning. So anybody who publishes a paper on STDP is by definition working on learning. No, the new bit is just let's just change what comes into the input, temporally structured patterns. And the stuff with Toby Delbrooke's chip is just a very neat way of generating highly structured temporal patterns. It's very difficult to understand if I was to just dump all the data on your desk. So, tell me what's happening here. Right? Because let me just point out the neurons, the connections between the neurons in Toby Delbrooke's retina and the neurons in that are totally unstructured. There's nothing. I could randomly connect, change the connections from the input layer to the output layer. It wouldn't make any difference. It will still pick up the same things because every neuron receives from 32,000 inputs, and it's just looking for a pattern of a bunch of spikes which has not been detected by somebody else. That's what they're doing. So there's zero constraints, which is why I think the potential is really very, very big, because if you can solve a totally unstructured problem in signal processing with no constraints, then, you know, I'm prepared to bet that this could actually, you know, a guy like you want to do, biosonar or, you know, whatever. If you put that into spiking temporally structured data and see what this sort of network will do with it, then you might get quite a long way. So, does it really happen in the brain? Well, I must give you, this is not in my presentation, but I think it's worth mentioning. We've been doing some experiments with Daniel Prestitz and Trevor Agus in Paris, where we try to sort of do this sort of learning with humans. So we, rather than using these spike things, we actually used Gaussian auditory noise. Okay, and we had a task which is actually a very difficult task, where on each trial you get a 1 second of auditory noise, which either is continuously renewed, or the first and the second halves are identical. And the task for the subject is simply to say, was the first and the second half repeat? So here, for instance, trial 1, it's going to be 1 second of noise, and it sounds like this. Okay, you're supposed to say no, right? No repeat. Now I'm going to play you one where the first and the second half are the same. You're supposed to say yes. I'm going to play another one where the first and the second half are the same. For me, actually, just for information, that sounds very structured. Why? Because I've heard this stuff before. That stimulus there is what we call a reference repeated noise thing, because it's going to appear later in the experiment. Subjects don't know that, okay? They're sitting there and just sort of, their job is every trial they have to, well, that was a repeat. That was continuous. That was a repeat. That was a repeat. That's up. I'm just cycling through the thing there. When you hear it a few times, then you begin to hear that there is a structure. So the subjects don't know that anything actually, there's any structure in the experiment. Nevertheless, if you take their performance on repeated noise, which they only get to hear once, I mean, the D prime's pretty terrible. I mean, you're above chance, but it's nasty. You know, and the subjects don't like it. But for the ones that got repeated later on in the experiment, the subjects get a lot better. And in fact, this is performance across all the repeated reference noises relative to the other ones, and you can see that it's going up within 10, 20 frames, 20 trials. But perhaps even more surprising is that, in fact, the learning is sort of all or nothing, because in fact, some of the stimuli the subjects do very well on, and some of them they can't manage. And we haven't, for the life of us, worked out what the difference between these are. And there's something idiosyncratic about certain noises which apparently make them easier to respond to, but they're not standard across subjects. It's not that this one goes and then it's easy. Strange. But you know, we're getting up to sort of like nearly 100% correct here within 20 trials. 10, 20 repetitions, you've already got learning going on. How long does the learning last? So here's the first block. Subjects doing pretty well, they're up to 90% correct. They go away for 2 to 3 weeks and come back, and we test them again. The very first trial, they can still do it. So now these are noise things. There's no way you can go home and rehearse this. I mean, the only way you can do this task is online with the headphones on. You can't go home and dream about it. At least it seems to be implausible. So we know that these memories will last for 2 to 3 weeks at least. And the next thing we want to do is to get the subjects who did this 3 years ago back into the lab and see whether they, first trial, can they still remember the stuff that they heard a long time before. How invariant is this learning? Well, if you speed up the audio file and play it twice as fast, it's still a lot better. Even at 4 times the normal speed, you're still a lot better. So it can support warping of the thing. It's not precise things. It's if you imagine neurons firing away within the stimulus, they don't really care what speed you're presenting it. Okay? And here's the killer. One of the reviewers said to us, well, what would happen if you played the thing back to front? This is what happens when you can play the thing back to front. You're still way better. You've never heard this stimulus before. It's just noise being played back to front. What on earth is going on here? Well, if you remember what I said about this thing here, this neuron, if you play the thing back to front, will fire at the end of it. So, it actually makes sense. If we were tuning up neurons that were selected to particular bits of noise, it would make sense that played back to front, you'd still have your string of neurons and just be firing in a different order. So it would still sound more meaningful when you hear the thing played back to front. Can STDP learn this? Now this is the latest stuff. This is Olivier Bichelet did the following. He took an auditory nerve model, very simple one. We just added a bunch of filters. There were 64 channels actually, and we just played noise into it and then generated spikes. So this would be a sort of typical thing for a 1 second of noise. 64 channels. So this is a pretty low data rate thing relative to the 32,000 channels in the visual stuff I showed you before. And then we feed that into one neuron, okay, with STDP, leaky integrated fire. And it's a bit complicated, but so this is the neuron. This is what it is firing away spikes, about 2 spikes per second. This is continuous, non-renewing noise. So it's going continuously, and the neuron's firing away merrily. And it will do that. We are satisfied that it will do it the way we set up the learning rule. When it fires spikes, it's reinforcing some synapses and depressing others. But on average, it's not shifting around. So it will happily fire spikes indefinitely. Okay. Then we start inserting a fixed bit of noise at random intervals. This is the first 4 seconds. First 20 seconds, sorry, of where the noise is coming in. This is, so 2.5 minutes later, then the neuron is only ever firing when the noise that repeats is occurring. It's mixed in with continuous noise. I mean, you would have a pretty damn difficult time trying to know that there's anything going on at all. But this neuron has picked up on it. And it's now firing is super selective. It will only ever fire essentially if that noise pattern is presented. And you can see its spontaneous firing activity is dropped down to 0, and this is back to the original condition, just continuously refreshed noise. And we let it run for I think several hours of simulated noise, and it never fired a single spike. So we essentially made a grandmother cell for this particular noise pattern, which is nothing special about it, in a few tens of presentations. And the neuron is sufficiently selective, it has 0 spontaneous activity and would never unlearn that stimulus unless you presented some noise pattern that was sufficiently close to get it over the threshold, and it might get tweaked then. As long as it's quiet, then it doesn't learn, then it won't forget. One neuron, and we can do this. And if you've got lots of neurons, they'll be learning, and you have speech coming in, or dolphin noises, or whatever. I mean, it's going to divide up the space into different neurons and so on. So, in the last remaining thing, I just want to say, well, I think this could be a secret to very old memories. Just for a bit of fun, I'm going to play you some clips from things you might have seen before, possibly. So, how about this one? You're very good at this. You obviously have the same references as me. That was, that was originally broadcast in '66 to '73, but it's been reached on. There's probably a TV channel somewhere that's showing it right now. What about this one? bien-aimé in French, Bewitched in English. I don't know if there are any other countries, but pretty well known. That was originally broadcast 64 to 72. Again, it's been rebroadcast, but I think you could probably find somebody who hadn't actually seen it who would still, oh, that's the theme tune to Bewitched, right? What about this one? Is this the oldies? Well, Terry Sejnowski shouted out "The Lone Ranger" about half a second after that started. I mean, you know, he was like me watching that stuff in the early 60s. That's really old. That was originally broadcast 49 to 57. No, it was rerun on TV channels in the 60s. I don't think people bother to do it now. It's really a bit too dated. And this last one, actually, but it's in your book. But this is quite interesting for me.
Stand by for action.
Thunderbird.
It's not Thunderbirds.
We are about to launch.
Stingray. Actually, Stingray. It was the pre-Thunderbirds. Thunderbirds has been re-shown a lot. Stingray, as far as I'm aware, hasn't. So I'm always on the lookout for some old Brit who was around in the 60s who might have not seen Stingray since then. That opening sequence, I used to watch it all every week. So I mean, I'd seen that particular sequence, several tens of times at least, maybe 100 times, because I've probably watched most of the program. And I think that's produced some pretty selective neurons in my head. So the long-term memory challenge is how to explain these very long-term memories. And my solution is that, well, if you believe all I've been telling you about spike-time dependent plasticity, how we make a neuron that's within 20, 30 presentations that's selective enough for an auditory noise pattern that it won't fire to anything else and its spontaneous activity is dropped out completely. That cell, if it's got STDP, would not be modified, for decades because it simply never fires. So I'm not going to have enough time to go through this, I don't think. But basically, there's a bit here that you can read through on neural representations. Well, I mean, do I have 5 minutes? Mm-hmm. But basically, there's the story here, and this is just a comment on what we were saying earlier on. I mean, you can have all the representations you want. You can have distributed, dense, sparse, and so on. They can have them all mixed up together. I'm not claiming that all the representations are these grandmother cells. You can have your distributed things where a high density of activity for any stimulus. There's a nice illustration from Peter Foldiak on the fact, here we've got 10 stimuli and 10 cells, and we've got the whole range. We've got local, sparse, dense coding, we've got grandmother cells, narrow and broad tuning. They're all in there. You can have everything at the same time if you want. So the argument is not to say that there's one coding scheme and that's all we've got. But I would claim that there's the Jennifer Aniston cells show that there are cells which have very high selectivity in humans. I won't go into those details unless you're interested. My last point is that those hippocampal cells are not grandmother cells, actually, quite clearly. And I won't talk about this, even though it's pretty simple. Well, the last point I want to make is that we have a fairly simple way of making grandmother cells. It's just STDP and sort of inhibition and so on. The question now, and the last point for discussion, is it possible that we have lots of cells which are like that cell that learnt the auditory noise and have just lost all their spontaneous activity? Well, the fact is we don't know. And the reason why we don't know is because when neurophysiologists record from, let's say, visual cortex or any cortical area, and you traverse 2 millimeters of cortex with your electrode, and you record every single cell that you meet on the way. That's how neurophysiologists like to do their experiments. I've done plenty of that. You basically will probably open a bottle of champagne if you get more than 10 cells. It's most of the time you'd be happy if you got half a dozen. Now, back in 1968, a guy called Dave Robinson calculated, on the basis of the electrical properties of a typical tungsten and glass electrode,
that you should be recording from cells within a range or a radius of around 50 microns. In fact, you can see this. If you go through a cortex, you start picking up your sound, it gets louder and louder and louder, and then it gets softer and softer and softer. That distance is about 100 microns. And David Robinson said, well, given the anatomy of the cortex, we should record between 100 and 220 neurons on every track, not half a dozen or a dozen. And that he sort of said, well, there's something fishy going on here. I don't know what it is. He sort of vaguely raised the possibility that it was maybe that lots of the cells are not firing spikes. But it's been stuck under the carpet since 1968, as far as I can see. There was one paper a few years ago that talked about this problem again. But it's perfectly possible that we have completely missed something really vital about brains, which is that we might have loads and loads of cells which you just never hear about if you're a neurophysiologist. The fact is that there are methods now which would allow us to answer this question. There's optical imaging and things like this, and implanted arrays, but I don't think anybody's really been asking that question. But it should be possible to get an answer in the coming years. So I would suggest that everybody keeps an eye open for people who can say something about what percentage of neurons in your cortex are currently active, and in particular what you call their lifetime sparks. What is the probability that neuron will fire a spike over the next 10 years? Is it 1 a second, 1 a year, 1 every 100 years? We just don't know. Okay. So end of story. I think that the extremely long-term memory thing is a major challenge. I don't know of any alternatives at the moment. That's good. Yes.
We can know, right? Because we know roughly how much energy is consumed per spike. We know the quiescent energy consumption of a neuron just to maintain it. And so by sort of measuring energy coming in, we could probably figure out the percentage of...
you're right. I mean, spikes are expensive. And
I think that has been done. So this was done by... it's embarrassing, I don't remember because it's a former colleague at NYU, but he did this calculation. It was actually a dean of some kind. And I think the answer was something like 2 or 5%. So the average firing rate of individual neurons in the brain is about 2 to 5% the maximum firing rate lifetime.
Okay, so that's the average firing rate.
Right, and I'm not
sure. That doesn't tell us whether there aren't half the cells which are firing at twice that, and the other half that are firing at 0.
That's possible. Yeah.
So you're right. Spikes are expensive. You don't want to do too many of them. And you can use the metabolic arguments to work out what the average number of spikes being generated per second is. But the average doesn't tell you what the distribution is.
Right.
So no, we don't know. We really don't know. It's perfectly possible that you just have to have, as I say, half the cells firing twice as much, or 10% of the cells firing 10 times as much, and the rest of them could be completely spikeless.
Right.
Except when Stingray comes back on.
So you would think that for efficiency reasons, for kind of, if you want to sort of have efficient representation or efficient use of, the representation, you would want the distribution to be more or less evenly distributed, except that we know for a fact that algorithmically or computationally, it's easier to learn things with very, very sparse representation. So for example, you can learn individual events or individual templates with template matchers. And template matchers, grandmother cells, are very sparse. But this kind of memory is thought to occur in the hippocampus or places like that. And so perhaps the hippocampus is kind of the kind that you're talking about, where neurons only react to kind of individual...
Neurons in the hippocampus, it's a Jennifer Aniston cell, they can be very, very selective, but the trouble is, I didn't mention it, but it's on the overhead. They're not grandmother cells because they're far too easy to find. If a real grandmother cell, you'd have to show 100,000 different pictures to have a hope of hitting it. The cells in the hippocampus, if you stick an array of electrodes in your epileptic patient and you're recording from 50 cells, let's say, which is what they do, and you then show 100 pictures 5 times to test it, that's what we do in the morning,
and you discover that on average you get a couple of cells which will be very selectively responding to one of those stimuli. Then in the afternoon you go and download another 10 pictures of Jennifer Aniston or another 10 pictures of The Simpsons or whatever, and you test them with that. But that just simply won't work, and that's what the authors say. These aren't grandmother cells because the hit rate is way too high. So something else is going on there. It could be, for instance,
cortical neurons, right?
no, it is no hippocampus.
Sorry,
That's not
hippocampus
corpus. That's not cortical. The hippocampus could be keeping a track of things that you've seen recently, for instance. That would be one option. I have a bet on with Itzhak Fried. I said if you've got a Jennifer Aniston cell here recorded one day, if the person watches Friends every week for the next 2 years, it will stay a Jennifer Aniston cell. If, on the other hand, Friends is no longer shown and they're not interested in it for 6 months, then that cell will probably end up doing something else. That's sort of the scenario. So in the human neocortex, there are almost no recordings whatsoever. So we just don't really know.
The point I was trying to make is that if you want to do one-shot learning, so just remember a particular event, then something like a very selective kind of grandmother cell like that just remembers the same, just turns on for this particular event, makes sense. So, for example, computationally, if you want to train a system to recognize a particular object, you use nearest neighbor. Nearest neighbor is just template matching, essentially, right? If you want to use something more sophisticated that has invariance and more complicated behavior that can recognize an object under various variations, then you need to do something that's more complicated, which possibly does not work just one shot. You need to kind of train.
It's not one shot; it's sort of 20 shots. You don't store absolutely everything you see. You store things which you are attentive to 20 or 30 times, and then you maybe learn, like the auditory noise, which one presentation won't get the effect. It ramps up over. Although you can get flashbulb memories as well, which is maybe something different, like a sort of thing where you sort of repeat internally the pattern.
I just wanted to point out that it seems to me that there's a trade-off between representational power and robustness to noise. So, for instance, you have n neurons, and if you use a dense code, you can encode something like binary dense code. You can encode 2 to the n representations, but only n representations with the localist grandmother cell thing. But if but the localist code has been deemed to be not robust to noise, in the sense that if this unit that codes for the stimulus gets down, then you're done.
So the claim is not that we only have one cell for each object. You can perfectly well have 6 cells for each object, and therefore the probability of all 6 of them dying, because you lose 100,000 cells every day, so you're going to be forgetting things. But if you have just a little bit of redundancy, and in a sense it would be impossible actually to make a system where I've got one cell responding to Jennifer Aniston. How is that cell going to stop everybody in the brain doing the same thing? No, it's not possible. It can stop its neighbours by division, but you're probably going to have, for things that you see a lot, you're going to have multiple copies in various bits of the brain, and so the problem of cell death being a major obstacle, I think, goes away. And in any case, we do forget things. I can't remember what the colour of my door when I was living in it. I can't retrieve that sort of information. It's gone. I'm not even sure. It would shock me if something changed about that now.
So would you say that this kind of redundant grandmother cell coding would explain why you can actually find very, very, very selective neurons in the brain, even though strictly speaking, the strict interpretation of grandmother cell coding will seem to suggest that this is impossible.
So tell me why it's impossible.
Oh, well, I thought you did it. I thought you mentioned that just before. That
in the hippocampus, it's impossible. In the neocortex, nobody knows, because I'm perfectly at liberty to say
that if we were to start recording individual neurons in the human brain, I mean, imagine now, let's imagine that I let somebody stick an electrode in my language area or something like this, and I'll just read the dictionary, and then I discover there's one cell that only responds if the word telephone is mentioned or something like that. Why not? It could be. Nobody's done the experiment.
If this redundancy can explain how the brain can recover from cerebral accident, for example, because in this case, the cell not to be retrained, but just to make the connections to reactivate maybe this hidden copy somewhere. No.
Well, the fact is that you don't let the memory off. So if you also take the network that learned to count cars, right, and I remove one of the cells that counted later before, well, another cell will take over because there's nobody covering that thing. So the thing will cope pretty rapidly and fill in the gaps. The rule is, you always want to have some neurons firing somewhere to code things. If there aren't neurons, then the inhibition is released, and that allows cells to start firing during the pattern and learning the pattern and so on. So the thing is very plastic, and it will cope in a pretty robust way, even if you remove things. And of course, the other thing is, one of the nice features of this sort of memory is that we don't say, I want this cell to learn about the Mona Lisa. You put in all this stuff, and you've got 1,000 neurons, and one of them happens to be to get selected for the Mona Lisa, another one gets selected for something else, but you haven't said which one has to do it. And that's deep belief networks maybe a bit, because you haven't told it what to do. It's just generated its own internal structure, and then obviously afterwards, when somebody says, "Oh, that painting there is the Mona Lisa," bang, you just make the connection, and then you've got it, because you already had a representation just by the visual experience. And I think this is maybe relevant. I haven't quite worked out what the relationship is with the deep belief networks, but I think the idea that you let the system use the data to generate its own internal representation, and then you label that afterwards, is a totally radically different way to saying, I've got my network, I'm going to use backprop to train all the internal things using by labeling the outputs and forcing the system to do a particular solution. I'm looking at Jan here, and I'm wondering whether you're going to say something.
Well, I guess, it depends what point of view you come from, but the idea of deep belief net is just deep architectures, and then the question is how do you train them, and numerically it's difficult to just train them with backprop unless you use things like convolutional nets. So if you put enough structure in them, you can train very deep networks purely with backprop. That works just fine.
So I would say if you've got to start it off with a 10-layer system,
right,
and you start putting in visual patterns at the bottom, first layer will form simple cells and complex cells, second layer will corners and stuff, third layer will do letters, fourth layer will do bigrams and trigrams, fifth layer will do, and nobody's said anything yet, and it's learning about words and stuff, and monkeys do this, right? That's we were talking about this at lunchtime. It learns about the statistics of what repeats. And then at the end of that, when somebody says that word means cat, and you make the association, then you 90%, 99% of the work's already been done.
Right. So funny you mentioned that, because there was a recent paper published at SCML by from Google that includes one of my former students, Marco Lorenzato Buckley, who's from Andrew Institute and Andrew. And they trained. So they used a sparse autoencoder, basically an algorithm that we developed, and they trained a sort of, you can think of it as a deep convolutional net, except there's no weight sharing. Just because they don't have the code for it. That's
oh, well, you're talking about the Google stuff.
Yeah, right. And so they train on YouTube video, and what they have in the end is basically a cat detector, because there's so many videos with cats.
Yeah, it was face detectors and working very well with cats.
So I thought it was a really interesting paper. It's actually, in a sense, it's a bit similar to the thing I did with Tim Maschelia. We put in the face database. We did use weight sharing, and it was very fast, 100 trials, and you've copied your face detectors all over the place. Now, the Google thing, they didn't, and it was obviously took a lot more computation to do it, but you end up with the same thing. You end up with that every bit of the visual field you get representations that will cope with faces over here, faces over there, cats down here, and so on. It requires more training because you can't just simply copy your weights across the map. But I mean,
well, it was relatively small images, roughly 150 by 150 pixels. So it's not, and they eventually applied it for recognizing objects in the ImageNet dataset, which basically have one single object centered in the image. So it's not like they were trying to do detection. So there, the weight sharing is only mildly advantageous, and it's only advantageous at the lower layers. The higher layers, of course, are sort of more global. But the reason they didn't use weight sharing is simply because they didn't have the implementation of it. There's no good reason for it.
Well, I personally would have difficulty in working out how the brain could do weight sharing. I mean, you're saying if I train to recognise a particular Chinese character over here, and I want to be able to recognise it over here, I don't know how to do it. I mean,
so I don't think the brain needs to do weight sharing for two reasons. First of all, because the hardware is already there in the first place. The brain cannot use hardware multiplexing. It has to have one physical neuron per virtual neuron, if you want.
Yeah.
And so, it would be more difficult to do weight sharing. Second of all, you can't recognize anything unless it's foveated, essentially. I mean, you can detect faces anywhere, but reading, you really have to foveate to read. And,
sure,
and so the fovea is relatively small. It's about 500 pixels on the side. It's really not that high resolution. And then the rest of the retina is, has this kind of geometrically decreasing resolution as you go away from the center. So you can't really use weight sharing there because because the statistics are so different, and you have more motion sensors on the side, and more color in the center, and more less color in the slight periphery around the thing. So it's not homogeneous at all.
But I mean, if you buy the story that maybe 20, 30 presentations are enough, how long does it take to have a face shown in the baby's visual field over here 20, 30 times? I mean, by the time it's a few weeks old, it will probably have had it. And if it was, if you had somebody actually talking over here and doing something a bit interesting, then in a minute.
So this, I think, I can't recall his name now, just now, Salk Institute, UCSD. No, with a Spanish name. I can't remember his name. Who tried to kind of do experiments with very simple shape recognition algorithms. Essentially, what amounts to sort of a two-layer convolutional net. So you would have, very simple filters at the first layer, maybe oriented edge detectors. There would be the pooling, and then just a linear classifier. And you try to figure out, how long would it take if you expose the system to images that a typical baby would look at. How long would it take for it to develop a face detector? It's basically minutes. I mean, it just goes like that.
Yeah, another amusing thing was somebody put a toy, a doll in a pram in the middle of a shopping mall somewhere, and programmed it so that it made a sort of baby noise, and then took a photograph 5 seconds later. And that worked very well because the baby makes a noise, people will peer over, and you could actually use that to babies make noises. People look at the and you're away. You can get face learning very rapidly.
Well, also the hardware in the baby is biased towards looking at things that are basically just 1 to 2 meters away, and so the only thing that is focused is face.
And there are these things that 3 minutes after birth, 2 blobs in a bar, and the baby will fixate it. So it's quite possible we actually might have something really basic wired into the colliculus or something. I don't know, some bit of the brain which just blocks the vision when that particular pattern arrives, and that would also help getting the sort of saccading to the face, or turning to the face reinforced. So you get a reinforcement signal if you got that, and here's a stimulus. I'm turning my head. Well, that was good. Reinforced the circuit.
So I don't know about you, but I'm extremely suspicious of claims of things being detectors like this sort of being there at birth. For the reason that,
yeah.
you know, even in our stupid algorithms that can learn oriented edge detectors, the real-time equivalent of what it takes to learn oriented edges is literally seconds to minutes.
I'm very sympathetic, except that you have to explain why everybody, or when many people are terrified by snakes and spiders. We do have some, you know, inbuilt aversion to particular types of motion, which is totally unjustified unless you live in Australia. The probability that a spider has ever done anything nasty to you is like zero. And it's not because people say, oh, there are some nasty spiders in Australia that you're going to be frightened of them. And yet people, there are, I don't know what percentage of the population is totally,
Arachnophobic, yeah.
Everybody here, worried that there might be a spider that might be hiding behind a lamp or something. This you can't explain that by rational, STDP and sensory input. I mean, there are a few things which are sufficiently important that it's worth wiring the visual system up so that you don't touch things which have that type of motion. You will stop.
Quickly, or did you conclude?
Oh yeah.